Inositol phosphates and phosphoinositides are metabolites that have several regulatory functions in eukaryotes, including the control of gene expression, protein trafficking, signal transduction, and cell development. They perform this regulatory function by binding to proteins, thereby changing protein conformation, catalytic activity, or protein interactions. The identification of proteins that bind to inositol phosphates or phosphoinositides is essential to understand how these metabolites perform their regulatory function. This protocol has a simple workflow that's sensitive, non-radioactive, liposome-free, and uses reagents that are commercially available. In this method, affinity chromatography with biotinylated inositol phosphates or phosphoinositides is used to isolate interacting proteins that are then identified by Western blot or mass spectrometry.
For bloodstream forms, grow T. brucei cells to mid-log phase in HMI9 media supplemented with 10% FBS at 37 degrees Celsius with 5% carbon dioxide. Keep the cell density between 800,000 and 1.6 million cells per milliliter. When ready, centrifuge the cells at 1600 times G and at room temperature for 10 minutes. Discard the supernatant and gently resuspend the pellet in 10 milliliters of PBSG, preheated at 37 degrees Celsius, to wash the cells. Centrifuge the cells at 1600 times G and at room temperature for 5 minutes to complete the wash. Repeat this washing procedure twice more. Then, resuspend the pellet in 1 milliliter of PBSG. Transfer the suspension to a 1.5 milliliter tube and centrifuge at 1600 times G for 5 minutes. Discard the supernatant and resuspend.